All right. I guess we'll probably get started now. We'll let everyone else join in. Anyway, these sessions are recorded and they will be available right after IWBDA, so folks can definitely catch up. So with that, good morning, good afternoon, good evening to everyone who's joining us today. My name is Prashant Vaidyanathan, and I work as a senior researcher at Microsoft Research in Cambridge, UK. And on behalf of the IWBDA organizing committee, it is my honor and privilege to welcome you today to IWBDA 2021. Now, IWBDA has always had a very special place for me personally, because it's not only my first conference in biodesign automation, but it was also my very first conference in synthetic biology, or even biology in general. And it's been a fantastic venue where researchers come together, they discuss concepts, methodologies, tools for computational solutions for synthetic biology and genetic engineering. And IWBDA has had a fantastic and an incredible 13-year journey. And each year this conference has grown and evolved, and it's built on the success of the previous year. So it was founded in 2009 by Doug Densmore, who is my mentor and my former PI, Mark Rydell, and Soha Soon. And after 13 years, here we are today. This is the second virtual conference. That last year was the first time when we held it virtually due to the COVID pandemic. And this is I think this is probably the first time we've had a 5-day event that's filled with so many amazing sessions. And all of this, this is a great feat, like trying to organize such a big event, like an online global event. And it was all thanks to a fantastic team. And I really want to spend a minute to thank them, specifically the BioDesign Automation Consortium. They've been organizing the IWBDA for the last 13 years. And this year I had a chance to actually understand the amount of thought and care and effort that goes into planning these events year after year. And so just a huge shout out to Aaron, Natasha, Tracy, and Doug for helping us in facilitating this event. I also want to give a huge shout out to the core committee members. Mary, who's the program committee chair, Jenhan, the publication chair, and Alexis, who is our logistics chair. That's a new event. That's a new position we created this year because we needed someone to actually help us coordinate such complex logistics. And all three of them have done such an amazing job, such a fantastic work here. And this event would not have been possible without all their hard work. Also want to take a minute to thank our program committee. Most of you may have seen them. They've done such a great job in giving such constructive feedback and helping with the abstracts as well. So just a big thank you to all the program committee members. And this year, in lieu of local chairs, because we normally used to have local chairs when we had this event in person, we have virtual chairs. So I really want to thank the virtual chairs as well, who are playing a huge role. They'll be a point of contact, and they'll be really helping us make sure that all the events run smoothly. So just want to thank them as well. So with that, it's really my pleasure to kind of give you a highlight and some of the an overview of what you can expect in this year's IWBDA. So as I mentioned, this is the 13th International Workshop for Biodesign Automation. And we have some things are consistent. We have some new changes here. So this year, so I just want to very quickly go over them. We have 2 fantastic keynotes, and we have 20 talks. And this year we decided not to have a poster session because, especially in a virtual conference, poster sessions are always a bit difficult. So instead we replaced poster sessions with short talks. So this year we have 20 talks, which has 13 regular talks and 7 short talks. And this year we have 8 workshops, which I believe is like a record number of workshops. And really, this is really building on top of the efforts last year where a call for workshops was introduced. And that really helped us. And so this year we have 8 workshops and 1 short discussion. We also been coordinating with bioRxiv to create a dedicated channel, which I'll talk about. We have an ACS synthetic special issue for this year's IWBDA as well. And then we also have some points to cover about the socials. So, it's this year, it's really my pleasure to announce that we have 2 brilliant keynote speakers, Dr. Tiana Radivojevic and Dr. Thomas Garchowski. Now, Tiana has been pioneering some exciting research that brings machine learning into the field of synthetic biology. In fact, I believe that her art paper, the automated recommendation tool, not only introduced, but I think it also popularized and inspired the idea of using ML techniques and like Gaussian process modeling and Bayesian optimization for design of experiments. And I think it's really exciting to see that kind of work, like those 2 fields come together. So her keynote will be later today, and I'm really looking forward to that. Our second keynote is by Tom, and Tom's been a big part of this community for so many years, and it's so exciting to have him as a keynote speaker this year. Now, his lab focuses on exploring molecular and biophysical mechanisms that individual cells and groups of cells use to make sense of their world and to process information. But I think Tom's contributions extend much more beyond this, be it sequencing methods or advanced computer modeling or visualization of genetic circuits. Tom's had a great impact in the field. So I'm very excited for his keynote as well. And his keynote is going to be tomorrow. Now, as I mentioned, we have 20 talks. We've divided this into 7 different sessions that spread over 5 days. You can get all the latest updates, and you can see the details of each session to know which talks are in which session by going to the IWBDA website here, by checking out the program. And this year we also have 8 workshops. I'm very quickly going to go over the workshops as well. SBOL has been a huge part of IWBDA. Like almost anytime I think of IWBDA, there's always an SBOL workshop, and the community has grown so much, and so is the standard as well, and it's really evolved. And we are currently in the 3rd iteration, the 3rd version of SBOL. And so this year, the first workshop is going to be the SBOL workshop that's later today. I think it's a great place to kind of familiarize with the tools and the libraries that are available. The second workshop is tomorrow, and this is really a great story because BioCR and Pyler was a paper that I remember it was presented at IWBDA. In fact, it actually won the Best Poster Award last year, and the paper has been published. It's open. The tool has been open sourced, and it's a very powerful library to model, simulate, and infer parameters for chemical reaction networks. So I'm really excited to see how past abstracts and past talks have really evolved and grown, and now we even have workshops to help people get used to this. So that's really very exciting. Our third workshop, this is a short workshop for about 90 minutes, and I think it's very interesting because like I'd mentioned, machine learning is increasingly playing a huge role in the field of synthetic biology. And now we're seeing how so many new machine learning techniques are helping us understand and give us more insight from the data that's generated. So I think this is going to be a great workshop as well. The fourth workshop is, I think this is going to be a fun workshop. This is by Krishna, who's a grad student from my former lab, CIDR lab. And he basically pioneered the 3DOF tool, and he's going to show you how you can use some of the CAD tools and CAD frameworks that have been developed to help build and create DIY microfluidics. Our fifth workshop, I think this is just another great story like BioCR and Pyler. So Flapjack was also another talk that was presented in the past at IWBDA. It's been published, it's been open sourced, and it's a great paper. It talks about it's a data management system for synthetic biology data, and this tool enables researchers to store, visualize, analyze, and share genetic circuit data, and it aids automation of synthetic biology workflows. So it's really exciting to see such papers evolve and become into like proper workshops as well. Now, we're also very excited for the BMSS workshop. So this is the biomodel selection system, which functions as a hypothesis testing tool that's been developed to perform automated model selection and enable a more systematic approach to the model development and analysis. So this is going to be on Friday. I just want to very quickly note that this session is going to start at 2 p.m. British Standard Time on Friday, unlike all other sessions that are starting at 3 p.m. So just wanted to just mention that very quickly. Now, whether you've heard of SBOL Visual or not, you've most likely seen it almost everywhere in every single publication, and it's had such a profound impact in ensuring that we standardize the way genetic circuits are visualized. So it's a brilliant effort, and Tom and James, they've been pioneering this effort for so many years now, and they've created numerous libraries to help and visualize these designs. So I think it's going to be a great workshop. So it's definitely one to check out. And finally, we have another one that talks about principles of genetic circuit design, which personally is it speaks to me a lot, because this is something that my research focuses on. So in this, Hatem will provide an overview of the design process for synthetic genetic circuits, for like gene expression control systems, composition of genetic circuits from different classes of gene regulators, different CAD tools and modeling tools used in genetic circuit design workflow, common failure modes, and so much more. So it's really exciting to see that. We also have a short discussion session. This is going to be about 15 minutes to half an hour. This is going to be a guided discussion session, and it's really interesting because it's going to talk about this big collaborative project, which is trying to design processes and protocols to support a network of mini-biofoundries, and just to be able to support this and make it accessible remotely as well. So this is basically a collaborative effort of 2 iGEM teams that they're going to prototype the workflow, and it'll be presented along with a collaboration with Beckman. So it's definitely something to check out. This is going to be on Thursday. Now, beyond this, as I'd mentioned, we partnered with bioRxiv this year, and one of the main reasons was because we wanted to make sure that we give a platform and we give a venue where all the workshops, all the abstracts that have been presented at IWBDA, they actually have a place where there's a DOI that can be referenced, because there's always so much great work that's presented in these extended abstracts. So we've created a dedicated channel. So please, once you've uploaded this to bioRxiv, please do let us know, and we can add this to our channels as well. And beyond this, we're also going to reach out to the community, and we're going to try to add all the past versions of the IWBDA abstracts as well. So we really want to grow this channel, and I think it's really helpful because it doesn't preclude you from publishing the same work in any journal or conference as well. So we're very excited about this. On a related note, ACS Synbio has, it's been such a big part of IWBDA. Since 2012, after every IWBDA, there's always a special issue where folks from the workshop can actually go and can submit their work here, and this is something that is going to continue this year as well. So I'm very happy to announce that we're going to have a 2021 IWBDA special issue. The deadline for that is February 28th, 2022. So we'd really like to encourage everyone to please submit a paper to ACS Synbio as well. Now, moving on to the social section. Now, we've created a special hashtag, which we've already received almost 40 tweets that have been promoting this event via these hashtags. So I just wanted to let you know, if you want to share your experience, if you want to share anything about the talks, or if you want to tweet about this, please use that hashtag, and we'll actually be able to feature your tweet on the website as well. So that's one. And the other thing is, so this year for socials, I really miss having this in-person socials, but obviously because we can't have that, we decided to use this service called Gather. It's really fun. I've used it a couple of times before. It's a venue where you can really come together and you can meet people. There's like private meeting rooms. You can meet in public. And so we've created a dedicated Gather Town a venue for this. So you'll see it in the program. There are slots for the socials as well. So please join these sessions, and it's a great place to network and learn and talk to each other. And finally, I just want to go over some of the logistics really quickly. So we've created a dedicated IWBDA 2021 Slack. This is basically the same Slack that was used last year for 2020. So we just renamed it Slack. So please do use the Slack for all your communications. Now the password to all the Zoom links, in case the direct links don't work, the passwords for all the Zoom links are IWBDA 21. It's pretty simple, but you should also be able to find it in your Eventbrite emails. All the speakers, please do contact your session chairs via Slack. There'll be an email as well. This is just so that the session chairs know who's going to present, and please also contact the virtual chairs as well. They'll also reach out to you, but this is also so that we can make sure that the right folks are added as hosts for each session. And please do try to join the session at least 5 or 10 minutes before each Zoom meeting starts. We're going to make sure that the meeting is up at least 5 or 10 minutes before. Please contact any of the organizers, any of the virtual chairs for any technical assistance. And please do check out the website for all the latest information. Now, we're also going to have an award for the best short and regular talk. And this is going to be announced on Friday, the September 24th, during the closing remarks. So please stay tuned for that as well. And finally, so this is something I'm pretty sure that I saw this slide all the way back in 2014, and I just wanted to keep this as well because I think this is a great slide, and we've had this for all the previous IWBDAs as well. I think it's really interesting. Please do try to meet new people. Try to maybe form a collaboration if possible. Ask questions of the speakers. Try to engage with the community. Please do try to submit a paper to ACSN Bio special issue, and we'll also be sending out a participation survey via Slack and via email as well. So please fill that out, and please do give us some kind of feedback, and please do let us know if you're interested in being part of this and helping us organize future IWBDAs as well. So with that, please let me hand over to Andrea, who's the moderator and chair for session 1, and enjoy IWBDA. Thank you.